Hey class, hopefully you are doing well. Here is our respiration and lung lecture. In this one we're going to talk a little bit about anatomy. I'm going to try to breeze through that relatively quickly because this isn't an anatomy class, so I'm not going to be testing on that too much. Just a couple of specific things. I'll talk a little bit about airflow. I'm going to talk about some partial pressures. If you don't know what that means, we'll kind of get into Dalton's law, um, just a hair. Then I'm going to be talking about acid-base balance. Before that, I'll talk a little bit about the Bohr effect. And lastly, some adaptation stuff related to the lungs and respiration. Really, I'm saving this lecture for just about last because really not a bunch going on here, at least relative to other tissues. So certainly skeletal muscle is probably the most plastic. But with this, I do want us to know quite a few things. So just starting off, we're going to be talking about respiration and more or less two different compartments. So think about pulmonary respiration. So a big term for that is ventilation, so just the exchange of O2 and CO2. And cellular respiration, O2 and CO2, more or less going the other direction. So we're comparing basically with pulmonary respiration alveoli versus skeletal muscle mostly is what we're going to be talking about there. But don't lose sight of this. The primary functions of the lungs or the whole pulmonary system are for gas exchange between like where we're living, on the planet, and the body, because oxygen, right, it's really important for aerobic metabolism, so all sorts of interesting stuff going on inside of the mitochondria. And lastly, acid-base balance. So there's a whole chapter in your book about acid-base and effectively how we're maintaining pH. And in the very short term, the lungs are the best way to do it, and in the long term, the kidneys are. So that's something to think about. Like you can increase your blood pH, meaning make it get more basic if you just hyperventilate for a little bit. So us breathing out more CO2 has a function of increasing our pH, meaning kind of making us less acidic overall. So functions of the lungs, some other stuff right here. You can read this if you're interested. Now, one big word here that I've talked about a couple of times, in the last lecture I talked about simple diffusion, but here I just want to define it again. So it's the random movement of molecules from an area of high concentration to an area of lower concentration. So that's mostly how oxygen and CO2 are going to be moving. So just generally think about it like this. Like in our environment, we've got a lot of oxygen, like 20.9%, whatever. Like, well, I'll show you that coming up. And a low amount of CO2 in the air that we breathe. Now, our cells produce a lot of CO2, so high amounts of CO2 within our muscle cells and low amounts of O2 relative. So that's kind of what's going on there. So oxygen is going to be going into our cells, and CO2 is going to be coming out of our cells. And whenever I'm saying cells, I'm meaning skeletal muscle cells. Now, with the just kind of overall structure of the respiratory system, I'm not all that interested in you knowing all of this. So, the nose, all of those mucous membranes, the pharynx, larynx, trachea, all of that. The main things that I want you to know are alveoli. That's the main site of gas exchange. And I mean, there's, type 1 and type 2 alveolar cells and things like that. But that's not abundantly important for this class in particular. I will point out something interesting with, like type 2 alveolar cells here in a bit. But those of you that have taken other biology or anatomy classes, I'm sure that you've already talked about that. Now, here, the major muscle of inspiration, meaning bringing air into your body, is the diaphragm. So we need to know that. And in terms of expiration, at rest, it's more or less, so exhaling is more or less a passive feat, meaning that we don't use our muscles to actually do it. Now, whenever we exercise harder and harder, we actually do more active expiration. But the diaphragm is really important. So for those of you that are interested in how we know a lot of different things about how muscles function, the diaphragm is one of the main ones. So in rats, we look at gastrocnemius, soleus, plantaris. In humans, there's some gastrocnemius, quadriceps, but there's a whole camp of people that do a lot of diaphragm research and use that as a model to understand how muscles work. And actually, the author of our book, Scott Powers, that's kind of his main model. Some other things, so there's pleura, and really I just want you to think about that as more or less like a membrane encasing our lungs. And that not really much else that I'm too concerned with about that. Now here, major organs of respiration, you can look through that if you're interested, but I'm not going to have you label that on a test or anything like that. There's the parietal pleura, visceral pleura, there's the diaphragm right there, bunch of things going on here. Now, overall, our lungs are more or less split up into a conducting zone and a respiratory zone. So the conducting zone overall conducts air to the respiratory zone. It humidifies, warms, and filters air. It has these different aspects of it. So big thing here, the conducting zone, there's no gas exchange going on. That's really the big thing I want you to know. And the respiratory zone, that's really where the business is happening. The gas exchange, right? So the alveoli, and we have capillaries running along the alveoli, and in those capillaries, that's really where the gas exchange is happening, just it's in and around the alveoli. Now, different professors will talk about, like, if we spread out all of the, if we spread out all of the alveoli, it can take up the surface area of like a tennis court or something bigger, maybe. Those are interesting things to calculate, but I don't find it abundantly important for you to know that. Again, here, just the conducting zone, respiratory zone, and here's our alveoli. And just keep in mind that, well, I mean, like, there's a lot of them going on there. Now, with type 2 alveolar cells, they're kind of progenitor cells for type 1 alveolar cells. So if we damage type 1 alveolar cells, type 2 can kind of, I don't know, almost like a stem cell be transformed into the type 1 alveolar cells. Really interesting stuff right there. So whenever we have damage to our alveoli, like in pneumonia or different illnesses or whatever, then the type 2 alveolar cells can kind of aid in that. And something to understand is where we actually have a lot of our immune cells, so like a macrophage or, you know, a white blood cell or something, we have a lot of those in and around the alveoli because, well, if something gets into our alveoli and like infects us, like a bacteria, virus, what have you, well, it's really important to have that big line of defense right there. Now, a lot of times things don't get down that far because of mucous membranes a little bit further up in the conducting zone and all that, but just in case, all sorts of things like that. Now, here, let's look at the mechanics of breathing. So, two big things, inspiration, expiration. So, inspire, I'm not trying to inspire anybody to do anything great in their life. I'm just talking about, yeah, it's a stupid joke. I'll stop before I keep on going. So, inspiration, really all that this is bringing air into the lungs. Expiration is expelling air from the lungs. So this is more or less characterized by the idea of bulk flow. So movement of molecules due to pressure differences. So bulk flow, simple diffusion, more or less synonymous terms, at least in the way that I'm using them. There you can read about this. So intrapulmonary, that just means within the lungs. So something to keep in mind here. So atmospheric pressure. So this is at sea level, 760 millimeters of mercury. And that really just means the amount of pressure or amount of stuff in the air. So there's more stuff in the air around sea level, and the further you go up, say a mountain or, I don't know, to the moon or whatever you want to do with your life, there's less and less stuff. Now the proportions of all the different gases stay exactly the same, but there's just less molecules of, say, oxygen the higher you go. The air up there is less populated, but the percentages are the same. So something to just pay attention to here. So at rest, 760 lung pressure, intrapleural pressure. See right here, those are approximately the same as atmospheric pressure, so there's no movement of gas. So whenever we inspire, we use our diaphragm, we cause like a pressure differential where there is less pressure within our lungs. And essentially how we do that is by increasing the volume, like expanding our lungs. And then air will go in, and see 754, 758, it's less than what's outside. And then whenever we expire, kind of our chest will fall, and then the air will go out of your face, right? So that's effectively how that's happening. Now, within here, there's major muscles of inspiration and expiration. You know, the sternocleidomastoid, the internal and external intercostals, diaphragm. Really the main one for inspiration, diaphragm. That's the only one I'm ever going to really ask you about. Now, muscles of expiration. Think about this as all of the abdominal or core muscles. Now, these are really only active whenever we're doing heavy labored exercise. Like, keep that in mind. So here's a question. Do respiratory muscles fatigue during exercise? So it was historically believed that they really don't. However, current evidence suggests that they do, maybe. So with either prolonged or high-intensity exercise, so respiratory muscles may indeed fatigue. And do they adapt? Yes, a little bit. They have an increased oxidative capacity. With that. And there could be reduced work for breathing, so after we adapt for a while, like ventilation does go down a little bit. Moving on to this next idea is airway resistance. So airflow depends on the pressure differences between the two different ends. So P1, that just stands for pressure 1 and pressure 2. And the resistance, so more or less the obstruction. So airway resistance is dependent on the radius or diameter of different parts of the lungs overall. So whenever there is a higher amount of resistance, like say if there's inflammation, like bronchitis or whatever, then they will be inflamed and they will actually expand, and their diameter will increase because, well, edema, they expand, all that type of stuff. That can certainly happen. That happens in COPD and asthma. We could talk a little bit more about exercise-induced asthma, but really just narrowing of airways, it can be induced by exercise. There's also like allergy-induced asthma. There's a bunch of different ways how to think about this, and I'm not going to really talk about it too much. Just be aware of it being a thing. It certainly happens quite a bit in many individuals. Now here, we could talk a little bit about COPD, but really just read in the book and go over this if you're interested in it. I'm a little bit more concerned with what's going on, just exercise in relatively asymptomatic settings for us. So, all right, let's move into some of these terms that we need to be aware of. So, pulmonary ventilation, what this is. So it's just the amount of air moved in or out of the lungs per minute. That's what V stands for. So there's a couple of different terms. So VT, which stands for tidal volume. So that is just the amount of air moved per breath. Breathing frequency, so just the amount of breaths that an individual takes per minute. So volume times rate, that gives us the total amount of ventilation. Now there's two different aspects of this. So there's alveolar ventilation and there's dead space ventilation. So alveolar ventilation, that's actually the air that is reaching the respiratory zone and that we can more or less actually do something with. And dead space ventilation is really just air going in and out of the conducting zone. So within our lungs we have something called an anatomical dead space, and it's interesting to think about. It can certainly impact the ventilation quite a bit. Like if you have ever seen cartoons where individuals take, like cartoon characters like Bugs Bunny or whatever, will take a straw and like go underwater and have that straw go up like outside of the water. That's functionally extending their anatomical dead space. So it doesn't work if you've ever tried it. I mean, I have, so. But going back to this, one thing here, that capital A, the reason we weren't using a capital A before and say something like the Fick equation for VO2 max is because a lowercase a actually stands for arterial and a capital A stands for alveolar. So if you ever see those two, the case capitalization or non-capitalization actually does seem to matter a little bit. Now there's a bunch of different terms related to the lungs that I do think it's important to know. And if you ever work in, I don't know, somewhat of a pulmonary rehab setting, these are important terms. So like vital capacity, FEV1, all sorts of things like that. So let's just go through these and let's term them out really quick. So more or less just vocabulary words. So vital capacity is the maximum amount of gas that can be expired after a maximum inspiration. So biggest breath that you can take in, and then the amount of air that comes out of your face after that. Now whenever we breathe out, we don't lose everything. We still have some air in there. And that is termed by the residual volume. So the volume of gas remaining in the lungs after a maximal expiration. Now, total lung capacity, TLC, not the channel or the band. This is just the amount of gas, in the lungs after a maximum inspiration. So that's the amount total that you can have in. That's the amount left over. That's the amount you can breathe out. Now here, a couple of different terms. And some definitions along with them. So we have an inspiratory reserve, expiratory reserve, vital capacity, inspiratory capacity, functional residual capacity, all of these different things. You can look at it in this way if that makes it a little bit easier for you. The main ones that I really want you to be aware of are tidal volume, residual volume, vital capacity, and total lung capacity overall. So this is more or less kind of indicating what's going on. And this is an individual with 6 liters of total lung capacity, which is quite a bit. So Yeah. I like a vital capacity of around 5 liters, which is being shown right there. Um, that's that's actually pretty cool and interesting, I think. Now, there's this whole idea of spirometry, where it's just a measurement of pulmonary volumes and rate of expired flow. So you can diagnose certain things like asthma, COPD with these. The main one being used is something called forced expiratory volume 1. And what that stands for is the amount of air that can come out of your face with um, like forced exhale in 1 second. So this gives us more or less criteria for determining if someone has COPD or whatever. It looks something like this. If anybody has ever been tested for asthma or COPD, you'd be aware of what that kind of looks like. Graphically, this is more or less how that looks. Now down here is the normal, so forced expiratory volume in 1 second, about 4 liters, so the majority of the vital capacity. And in a COPD individual, there's a smaller ratio, so meaning that really the resistance, there's an obstruction in there, so it's harder to have a greater rate of flow coming out of the lungs. So there's some interesting things there if you are interested in that. Next stuff going on, a couple of numbers to be aware of. So the main things that are within our air are oxygen, obviously, CO2 to a quite small degree, and nitrogen being the vast majority of it. And there's a really small amount of, the noble gases like argon, xenon, neon, helium. Yeah, that's another one. And those always have the same fraction, and we can calculate overall their amount. Really the amount of particles of that in the air based upon what we know the fraction is and what we know that the barometric pressure is. So it's kind of hard to conceptualize barometric pressure, but here's how I think about it. It is on a gradient of the bottom part of the ocean to the highest point on a mountain. If you've ever gone really low, or I don't know, dove really deep in a pool or something like that, you feel a lot of pressure on you. And now, granted, it's a liquid that's compressing on you, but liquids and gases are really the main difference between those is how clustered the different atoms are together. And in gases, they're just further apart, so there's just less pressure overall. So essentially what that means is that the lower you are, the higher the amount of pressure, the amount of just air and particulate in the air is compressing down on you, and the further up you go, there's less of it because it's essentially under you. I don't know if that helps anybody understand this at all, but that's how I conceptualize it. Scaling back just a little bit, that's more or less how I explain partial pressure. Now, Dalton's law, this right here, is the total pressure of a gas mixture is equal to the sum of the pressure that each gas would exert independently. So essentially we have all of these different gases, and then we sum them together, and that's how we get the total barometric pressure. Now, the diffusion of gases, here's another equation. So this is Fick's law of diffusion. So not the Fick equation for VO2 max that we were talking about before, but if I refer to this on an exam, I will always talk about Fick's law of diffusion. So this is the rate of gas transfer. It's proportional to the tissue area and diffusion coefficient of the gas and the difference in the partial pressure of the gas on two sides of the tissue and inversely proportional to the thickness. So right here, V gas, that is just the rate of diffusion. So area, right, that's the area there. How thick the tissue is, the diffusion coefficient, which is likely a constant for whatever particular gas that it is. And then here it's just the partial pressure difference between one end and the other end. So overall, that's the main business that's going to be driving this, because we really can't alter much of the area of many of our tissues or the thickness of them. Certainly in clinical conditions, the thickness can go up. But really the pressure difference is the main thing that's going to be determining the rate of diffusion. So if we're looking at this in this way, let's pay attention to the two main gases that we're interested in, and that being oxygen and CO2. So what these are, oxygen, 159 millimeters of mercury, CO2 very low. In alveolar air, it's already dropping down. So it's essentially going to drop down this whole time, and then it's going to kind of pick back up over on this other side of the equation, or of the figure, sorry. So essentially inspired air 159, 104, then in the blood it's around 95, and then in the tissue it's around 40. So if you notice, this number is going down. And in terms of the CO2, it's essentially not changing, and within all of this, the CO2 is the highest within the tissue. And that being the highest in the tissue is going to drive it to leave in the venous blood, and then be exported out. So pay attention to this, 46 to 40 to 32. And really in the normal air, 0.3. So that's kind of the directionality of CO2. Hopefully that makes sense. O2 is going down, and then CO2 is going up in that way. And whenever I say CO2 is going up, I mean it's the highest here, and the partial pressure is going to be going down as it leaves the body at all of the different stages of it. So hopefully that wasn't too confusing. Just follow the numbers on that. So let's move on just a bit to the blood flow to the lung. So interesting parts about, there's going to be something called the VQ ratio. So overall, Q is going to mean the essentially like blood flow going to it, and the V is going to be the ventilation. So the pulmonary circuit, it has the same rate of flow as a systemic circuit, but it's a lower pressure. So when standing, most of the blood flow is at the base, due to gravity and stuff, right? And most of the ventilation is at the top of it. So during exercise, more blood flow gets to the apex. So here you can look at this if you're interested in it. It's more or less what we've been talking about this entire time. So here, regional blood flow, within the lung. So there's greater blood flow at the lower rib number, and there's less blood flow at the higher ribs. Yes, here, this is what I really wanted us to get to. So ventilation, perfusion relationship, so VQ. So this will frequently be called VQ mismatch. So ventilation, perfusion, really I want you to know that this is ventilation in the numerator, perfusion, or blood flow in the denominator. So the ideal rate is approximately 1. And overall with the lungs, there is a part of it that is underperfused, meaning not enough blood flow, and there's another part that is overperfused, meaning too much blood flow. So that just means that really the ventilation and perfusion is a little bit inefficient. And that's fine because at rest, all that's good. Now, during exercise, it certainly improves this relationship, meaning getting it overall to approximately 1, so we get essentially more blood flow to the apex and more ventilation in terms of a ratio to the base. Now, in heavy exercise, there is still the inequality there. So if we look at it this way, so at the apex, ventilation, strangely enough, I mean, like, there is more ventilation at the base, but it's just a very odd relationship with very low amounts of blood flow at the apex and really high amounts of blood flow at the base. And that ratio, it would be better if it was nearer to 1. So 1 or slightly greater, it's really, I just want you to know about the 1 right there. All right, so regarding O2 transport in the blood, in lab we've actually been able to measure this a couple of times with something called a pulse oximeter. So essentially what that's doing is it's sending infrared light to kind of through your finger to see how much oxygen is actually on hemoglobin. And hemoglobin has a very interesting protein structure, which I would like to get into, but I'm sure that most of you don't want me to. And that's fine. So 99% of O2 is transported by hemoglobin, which is notated as HB. The amount of O2 that can be transported per unit of blood is dependent on the hemoglobin concentration. So really just how much that you have. There's a lot of reasons why individuals say, well, blood dope or take something called erythropoietin, which erythropoietin is just a hormone that will more or less make it to where you have more red blood cells, and blood doping is essentially taking out the red cells, which have, erythrocytes or hemoglobin in them, and then putting them back in, and if you just have more of those, then you can carry more oxygen. And there's actually some research where individuals who are untrained just did a blood doping protocol. This was done a very long time ago, whenever people didn't care about ethics. And they just put more of that in them, and they were able to last quite a bit longer. Now, oxygen content of the blood, here it's 100% hemoglobin saturation. This math right here, I'm not particularly concerned with us knowing it. Now, we're going to be getting into some dissociation curves. So this is really just the rate at which oxygen will leave hemoglobin. And it's slightly different under different conditions, and this is going to be really important for the Bohr effect. How this works, deoxyhemoglobin and oxyhemoglobin. So oxyhemoglobin, whenever it dissociates, becomes deoxyhemoglobin, and that oxygen essentially is diffused into a cell where, it binds to myoglobin, and then, it'll be transported to the mitochondria, more or less. So the direction of the reaction depends on the partial pressure of oxygen. So how much oxygen is anywhere. So at really high amounts of exercise, the muscle cell or mitochondria is consuming lots of oxygen, so it's going to drop. And also, we're going to see some different things where the acid or pH will alter some of these things. So there's also different tissues, or not tissues, I mean different things that have a higher or lower affinity for oxygen, meaning they're greedy and less likely to give it up. So myoglobin has a much higher affinity for oxygen than hemoglobin is. So essentially that means that the partial pressure has to get really low for myoglobin to lose it. And I'll show you that here coming up. So at the lung, we have a high amount of PO2, so that's how we form oxyhemoglobin, and at the tissues, low amount of oxygen, so we release it. So at lower PO2 levels, the oxyhemoglobin will let it go. So essentially, looks something like this. So, we have like PO2 in the arteries and veins at rest and percent oxyhemoglobin saturation. So over here, well, percent oxygen content, you can look at that too. So essentially, it's more likely to dissociate. So oxygen content, it's going to come off, or percent oxyhemoglobin saturation, it's going to get less saturated the lower the PO2 is. And if you remember, within our cells, like our muscle cells, it's around 40 or so. So it's likely to come off, and we're going to get some oxygen off of the hemoglobin at that time. So here are three different things that I want you to be aware of. So things that alter the oxyhemoglobin dissociation curve are pH, temperature, and 2,3 DPG, which here I'll just start at the top. So pH, a decreased pH lowers O2 affinity. So it results in a rightward shift, meaning that it will come off at, well, here, let's just show you right here, meaning that it's going to come off at a higher PO2. So this is a bit counterintuitive. So this middle one right here is about normal. So if we get more acidic, as we would during exercise, the oxygen is going to come off the hemoglobin sooner. So that's a good thing, because whenever we have a lot of acid going on, that means we're exercising. It means we need to get oxygen into the cell and CO2 out of the cell. And essentially they're going to swap. Now, whenever we're really basic, meaning that, well, we're not really exercising, it's going to hold on a little bit tighter. Now, temperature does approximately the same thing. So increased blood temperature lowers the O2 affinity, so meaning that it's going to come off a little bit easier. Now, 2,3 DPG, this is measured during altitude. Most of the time, it really isn't a major cause, especially under most conditions, but it does still play a relatively large role. But here, something to pay attention to is this rightward shift. That is essentially oxygen coming off more easily. So hemoglobin is not going to as greedily hold on to the oxygen, more or less. Now, similar thing in terms of temperature. So our normal temperature is 37 degrees Celsius or 98.6, like whatever you want to think with that. Now, if we're starting to get warmer, 42, it's going to come off at a higher PO2, right? Now, here, oxygen transport within the muscle, myoglobin, or MB, has a much higher affinity for oxygen. So this is actually what shuttles oxygen from the cell membrane to the mitochondria. And so it has a higher affinity, even at much lower PO2s. So this allows myoglobin to store O2 and have like somewhat of a reserve amount of oxygen within the muscle. And really a big part of this reasoning is that within the mitochondria, I believe your book says this, that the partial pressure of oxygen can get down to like 1 or 2 millimeters of mercury. So it's really important for it to have this super high affinity. So here is more or less how this looks. Essentially showing that myoglobin, it's really greedy with oxygen. The PO2 has to get kind of insanely low for myoglobin to want to let go of it. Now, this insanely low amount of pressure of oxygen certainly does happen during exercise, so it's not like it'll never let go, but that's more or less the relationship there that I want you to know. now we're going to get into some fairly interesting stuff. So we just talked about how CO2, I'm sorry, how O2 is effectively getting into the cell. So oxygen, hemoglobin, lower partial pressures getting in there. Now there's an exchange of O2 and CO2, right? So how does CO2 get out of the cell? Three big things here that you need to know is how CO2 is transported in the blood. So it's transported by three different mechanisms. The smallest part is dissolved in the plasma, so there is just some CO2 circulating around in your blood. Another part is just bound to hemoglobin, and I'm probably going to say this incorrectly, but it's like carboxyl hemoglobin. Something like that is how the pronunciation works. You can look it up if you're interested. But the vast majority is actually going to be transported as bicarbonate. So that is effectively this equation right here. So CO2 mixing with water, carbonic anhydrase, and then essentially over here with well, like that's bicarbonate, and hydrogen being kind of happening going on there. So like here's a bit of a thing going on here, and there's a particular thing called a chloride shift that I want you to just know happens. So if we're looking at this picture, let's pay attention to it right here. So within a cell, CO2 is produced, as a byproduct of aerobic metabolism, right? So some CO2 just goes into the blood, some of it, hemoglobin, CO2, carboxyhemoglobin right there, some mixes with water, and then down the road a little bit we get bicarbonate. Now, bicarbonate is actually shifted outside of the red blood cell, which another term for it is erythrocyte. Now, to maintain this electrochemical gradient, because that has a negative charge, then chloride will shift into that cell to essentially make this more or less neutral in terms of like an electrochemical gradient. Now, a really big thing here, if we're taking this HCO3 and taking it out of the red blood cell, that means that we can actually get more carbon dioxide into here, and we can essentially do this more and more. Now, if this didn't happen, and that bicarbonate had to functionally stay within the cell, then we would have a relatively large problem of there would be a backlog, and then it would stop CO2 coming into the red blood cell. All right, so on the other side of this, and if this isn't making sense, try to slow it down and read it in your book. Whenever we get CO2, whenever we expire it out, breathe it out, functionally the equation goes the other direction. So the carbonic anhydrase equation will functionally go the other direction, so that we end up having water and CO2. So hopefully that is making some sense right there. All right, so going on just a little bit. So ventilation in relation to acid-base balance. So pulmonary ventilation actually removes hydrogen ions from the blood by this bicarbonate reaction. So in a sense, it being closely associated with hydrogen and the bicarbonate, it's really removing it and functionally making it water in many ways, right? So increased ventilation results in CO2 exhalation, so that actually reduces the carbon dioxide partial pressure, and the hydrogen ion concentration. So decreased ventilation results in a buildup of it. So different things that we can do, whenever we're huffing and puffing really hard, that more or less is trying to drive up our pH to lower the acidity so that we can continue with, normal metabolic functions. Now, let's go and look on to this a little bit, and I'm going to try to wrap this lecture up a little bit faster because really most of that was what I wanted us to be aware of. So in terms of rest to work transitions, at the onset of constant load submaximal exercise, there is initially a ventilation increase, then a slower rise towards steady state. So people will start breathing somewhat hard rapidly. Now, PO2 and PCO2 are relatively unchanged. There is a slight decrease in O2 and a slight increase in PO2. So right here, this overall suggests that there's an increase in alveolar ventilation, or that the increase in alveolar ventilation is slower than increased metabolism in many ways. So here, if we just pay attention to this, PO2, it's going to stay relatively the same. Now at max exercise, it's probably going to drop quite a bit, and CO2, that's probably going to go up. But overall, this this ventilation, that's going to go up a lot, and O2 and CO2 are really not going to change very much. Now, in exercise in hot environments, ventilation tends to drift upward, so kind of like cardiac drift, what we talked about in the last lecture, and there's really little change in CO2. So kind of this is what's happening here. So ventilation is going to end up going up, whereas CO2 is really not going to be moving around too much. Now, incremental exercise in untrained individuals, it seems to be a linear increase up to around 50 to 70% of VO2 max, and an exponential increase beyond that point. And this is called the ventilatory threshold. It's highly associated with the anaerobic threshold, the onset of blood lactate accumulation, the lactate threshold. All of these are really just different ways to triangulate and get to a particular point, right? Now, PO2 maintained within 10 to 12 millimeters of mercury of a resting value. Now, that's the untrained individual compared to an elite individual. So ventilatory threshold occurs at a much higher level of VO2 max, so much higher than 50, 75. So PO2 decrease of 30 to 40 millimeters of mercury at near maximal work. So highly elite individuals can actually plummet their partial pressure of oxygen in their blood quite a bit. So this is hypoxemia. So that just means low blood oxygen. Emia just means blood. Hopefully you're aware of that. And this is likely due to a VQ mismatch. But one thing that seems to make a lot of sense is something called pulmonary capillary transit time, meaning that blood is going so quickly through the pulmonary capillaries that there is not enough time for the red blood cells or hemoglobin to get fully oxygenated whenever blood is pumping through the pulmonary capillaries. Now here are just some differences between an untrained and trained individual. So PO2, untrained individuals don't seem to be able to plummet it that much, how compared to trained individuals seem to be able to really plummet it down, and that's probably mostly due to the pulmonary capillary transit time. Now the pH, there seems to be a almost an inflection point drop-off for a untrained individual, and not quite a linear increase in ventilation, but more so than in a trained individual. So there's just some differences there. Hopefully these ideas are making sense to you. Now, in control of ventilation, there's really just a couple of things that I want us to know here. So it has a lot to do with higher level brain centers, you know, like medulla oblongata, hypothalamus, all sorts of things. Here, let's see where I want you to get. So in the medulla and the pons, there's different things. Let's get through this a little bit faster. So there's humoral and neural input areas. So some things to really pay attention to that will increase or decrease ventilation. We are susceptible to blood oxygen levels, but actually our body pays a little bit more attention to blood carbon dioxide levels. So here, let's get to right here. This graph and the next graph are the ones that I really want you to be more aware of. So arterial PCO2, so partial pressure of carbon dioxide. It seems like ventilation is a lot more strongly tied to CO2 than to O2. Now, we are, once blood oxygen levels get down too low, that will increase ventilation, but it has to reach a threshold. And CO2 seems to be a bit linear, a bit stronger of a relationship. So this is kind of how you can increase how long you can hold your breath. So if you hyperventilate for a really long period of time, and well, whenever I say long, I'm talking like a minute or two, you can actually hold your breath quite a bit longer because you're clearing out blood CO2. So if you like hyperventilate quite a bit, you can drive your CO2 levels down really low, and then you're not going to get the impetus to breathe for quite a while. And that's kind of how like Harry Houdini and oh gosh, what's his name? David Blaine. If y'all are aware of different magicians and things, how they actually seem to increase how long they could hold their breath. That a lot of it was due to hyperventilation. Now, O2, less of a tie. We do respond to it, but it has to get to a fairly low level, and then it'll drastically increase. Here, ventilatory control. So it seems like during submaximal exercise, there is a primary drive from higher brain centers, and it is fine-tuned by chemoreceptors, so chemical meaning. And really what we're thinking about is CO2 and O2 going on. You can look into this heavy exercise and a linear rise in ventilation with that. You can check out some of this stuff, but I really more or less told you what I want you to know with that. Now, training, what are some training effects? A couple of things that I want you to know. So training reduces the ventilatory response to exercise. Now, big thing here. It's probably more due to adaptations within the musculature rather than adaptations to kind of the whole pulmonary system. So exercise really has no effect on lung structure. So physiologically, that doesn't really change much. Now, ventilation is lower during exercise following training, 20-30% or so if you get fairly highly trained. And the mechanism seems to be the aerobic capacity of the muscles. So producing less hydrogen ions, less anaerobic work overall, and less of a afferent signal back to increase respiration. So this is more or less what's going on here. Like before training, you'll breathe more during a specific workload or work rate. And just think about this work rate in watts as running at, I don't know, 5 miles an hour, 6 miles an hour, 7 miles an hour. Now, after training, you'll actually breathe less. So the liters per minute will actually go down quite a bit. And it's mostly due to adaptation at the muscles involved in whatever type of movement that you're doing. Now, the effect of training on ventilation, so no effect on lung structure again, and function at rest. So normal lungs actually exceed gas exchange. So what do people say? They say that we're over-respirated in a way. So adaptation is not required for lung to maintain blood gas homeostasis. Now, there does seem to be this one exception for elite endurance athletes, so they could have a failure of the lung to adapt to training result in hypoxemia. Now, overall, the main picture is not much of a change going on there. And then the question of does the pulmonary system limit exercise performance? Not really, and throughout most of history wasn't thought to be a limitation, but in elite endurance athletes it certainly could be, and this is likely due to the very fast pulmonary capillary transit time where red blood cells or hemoglobin don't have sufficient time to get saturated with oxygen, so that whenever they're circulating, they're essentially at a low oxygen level the entire time. So that was really the totality of this lecture. If you have any questions, please email me.